Hello and welcome back. Today we are diving into the perception of stimuli. Basically, this is going to include all of your eye and your ear and your nose, and how you can actually receive information from those areas and like have your senses work. So diving in, let's talk about the types of receptors that exist in your body, so that when I mention them as we go through the rest of these notes, you kind of understand what they are. So thermoreceptor is for temperature. This is how you can actually determine that something is hot or cold, but usually hot. Photoreceptor is for light. This is found really in your eyes, rods and cones, to determine like black and white color in dim light versus like color like yellow or red in bright light. So we'll talk more about rods and cones in a minute. Mechanoreceptors, they're like a lot of your typical receptors, like when someone touches your hand, you can feel that. You can feel pressure. Sound waves, if you've ever been to a concert and you can feel that bass like thrumming through your body. You have hair cells in your ear, which helps detect when you're moving and when you're not. Like, especially if you get like car sick, you'll know that maybe they're working a little too hard. And then baroreceptors in blood vessels, that's with pressure. And then chemoreceptors detect chemical substances, so you can smell chemicals. There's pH receptors in some blood vessels that helps maintain homeostasis. If you have an out of whack pH, those receptors will recognize that. Pain receptors detect chemicals released by damaged tissue and can start either like a blood clotting cascade, or just alert the brain like, hey, we're in pain, like please do something. So let's look specifically at our nose. So this goes from bottom to top. So there is some sort of odor in the air. Let's say it is the smell of cookies baking. So you are starting to smell like the fact that the chocolate chips are melting and the butter is oozing, right? So you are getting these odor molecules flowing through the air, and they will bind to receptors that are found within the nose. And those receptors are called olfactory receptors. Olfactory is just like the science word for scent, for smell. So like your olfactory nerve deals with your nose, olfactory bulb. You'll see like all of it is just smell. So those odor molecules bind to the olfactory receptors. Those receptor cells are activated and send an electrical signal up to the glomeruli, and that is like within the olfactory bulb of the nerve. And so these receptors send signals to the nerve sends signals to the brain and says, hey, I smell cookies, like let's go get some cookies, right? So the odor molecules bind to a receptor and the receptor sends information. Pretty straightforward. Now, structures of the eye, I would just pause and maybe draw this. You should be able to draw and label an eye. The functions of the eye are, I didn't put them in this set of notes because there's like so many, right? We're gonna like spotlight a couple, but there's just so many. We are going to do an eye dissection, in which case you will write down all of these like functions and what's important, etc. But if you're looking right now for the functions of the eye in our textbook on page 524, there are all of the functions of these, but for right now in your notes, what I would like you to do is just draw this and label it. So like I said, we were gonna like spotlight a few. One is the lens. The lens is like a thick It almost feels like a marble. It's not perfectly circular, it's like a flattened marble, but it's thick and it's hard. It's like you can, not that like I suggest you like take out your lens or anything, but you can like kind of like knock it on a table and it makes a sound. It feels almost like a slightly squishy piece of glass. It is responsible for focusing the image, whatever image it is that you are looking at, for the retina. And the retina is along the back of your eye. So you see an image like this R, and your lens inverts the image and reflects it onto the retina. And then later on, your visual cortex in your brain reverts it back to its normal, like, correct side up. Your body's weird. Like, I don't know why it does that, but it does that. And then the blind spot is where the optic nerve meets the retina. So you can see it here, the optic nerve is there. When the optic nerve actually joins into the retina, there are no photoreceptors. So any light that falls onto this blind spot is just not perceived. Everybody has a blind spot in their eye. The rest of your eye kind of makes up for it, so you don't really notice that you can't see out of like a small section of your eye because there's so many other receptors and your eyes work together to kind of counteract any blind spots that may appear, but it does exist. And then the retina itself, I mentioned rod cells and cone cells earlier, but rod cells allow black and white vision in dim light. So rod cells would be like when you're driving in like the early morning or like dusk to evening when it's like kind of dark out, but it's not like pitch black out, but you know, the street lights are coming on and it's just dim enough. Rod cells allow you to actually make out your surroundings in that dim light. And then cone cells allow for color vision and bright light. So that's like during the day you can see like colored paper, like stuff like that. So in this case we have our photoreceptors. These are rods and cone cells. These are then sending impulses to the bipolar neuron cells, and these carry the impulse to the ganglion cells, which are right here, and then the ganglion cells send impulses to the brain from the optic nerve. So it's a very similar setup to scent and how your nose picks up whatever it is you're smelling, but instead of odor molecules, you're getting the direction of light. So you're seeing the reflection of light off of your retina. So, perfect timing. I didn't realize this was the very next slide, but the perception of light. So, in this case, we have light coming in. You can see the arrows. Light is coming in, and it's bouncing off the choroid and hits rods and cones. Okay, so it comes into your eye, bounces off the back, and hits your rods and cones, wherever they may be in relation to like the light. The pigments in rods and cones break down. Okay, you're kind of breaking down what you're seeing. Stimulates an action potential, which is the ability to send an impulse through your neurons, and the synapse passes that impulse to the bipolar neurons. So we're like starting the message. We're starting to send the message by stimulating an action potential. And then once you stimulate that and you pass that impulse on to the bipolar neurons, it goes into the ganglia cells and they will carry the impulse to the brain via the optic nerve. So this is just like another visual and wordy breakdown of how we actually perceive light. So there are 3 types of cones that you should be aware of. There are red sensitive cones, green sensitive, and blue sensitive. It just allows for the specific colors that you are able to actually perceive. These are the cones that are influenced with people that have color blindness. If you're red-green colorblind, then you have issues with your red and green cones, where you can't differentiate between the two. The question on the bottom, which cones would be activated to allow us to perceive yellow? It's anywhere that you see yellow, you would have those cones activated. So in this case, there's some red, more green, but both would be activated to actually see the specific color yellow, whatever yellow it is that you're seeing. And then your visual field is an interesting way, like, so we talked about it earlier, when the image that you're looking at is actually reflected onto your retina, it is inverted. But also, light from your left visual field falls onto the right side of the retina and vice versa. So you see that it's crossing, so your left visual field in the left eye goes onto the right side of the retina, and the right visual field goes onto the left side of the retina in the left eye. So it is like crisscrossing at the lens to reflect on opposite sides. And then like same with the right eye, but like opposite. So color blindness, like I was just talking about in the slide or two ago. So human vision uses trichromatic, is trichromatic, right? We have 3 classes of cones. We have the red, the green, and the blue. Dichromatic is basically color blindness. So if you lack red cones, you have red color blindness, or green cones, green color blindness. You have this issue due to an absence of or a defect in the gene for photoreceptor pigments. So this is a genetic issue. It falls on the X chromosome. It is sex-linked, which is why it's more commonly found in men than in women, because they only have 1 X chromosome, whereas biological females have 2 X chromosomes, so there is less likely of a chance to have 2 defective X chromosomes, whereas it's much easier to have 1, and then you would have color blindness. So again, it's a genetic issue, and it's messing with the photoreceptor pigments that would allow your cones to function normally. Okay, now processing of visual information, this is actually a very similar setup to specifically in the eye. So when you're looking at the eye, you see this like blue part in this picture and this red part. Remember that you are taking in from the opposite field of vision into your retina. So left field of vision, right side of the eye, right field of vision to the left side of the eye. From there, you have the optical nerve that is connected to your eye and to the optic chiasma. So this is where the left and right optic nerves meet. So from your right eye and from your left eye, whatever information is being processed through that optic nerve will meet up in this middle portion where like this big X is. This is called the optic chiasm. And the information that you are gathering then crosses over depending on where it's from. So you have information from the left visual field crosses over to go to the right side of the brain, and information from the left side of the visual field crosses over, no, left goes to right and right goes to left. So it crosses over, you can see it. So, right, we're starting in this like red area in this middle. It is from the left visual field, it is hitting the right eye on the right side of the eye, and then it crosses down, and this left visual field information goes onto the right side of the brain. This is, you're probably like, whoa, why does my brain do weird things? Well, to be honest, we're still figuring out a lot of that stuff. But in this particular case, this allows the brain to deduce size and distances. This helps with determining how far away something is. Is it a giant Godzilla monster coming to attack me, or is it a little like gecko climbing a tree, right? Distances, size, it helps you actually determine like field of view. Okay, so that was the eye. So we've done the nose and the eye. Now we're going to move into the ear. Again, I would pause this, I would draw this, and I would label this. This is another thing that you are just going to have to know. This is, unfortunately, just one of the drawings that you have to be familiar with, whether it is straight up drawing it from memory or labeling an already drawn image. I don't know what the test is going to ask for, but I just want you to be aware of what is possible. So please pause this and draw this. And now let's talk about how sound is perceived. So the middle ear bones receive vibrations from the tympanic membrane, which is your eardrum. The stapes strikes the oval window, which causes vibrations. So let's just go back for a sec. Okay, so tympanic membrane is right here. That's your eardrum. So vibrations go into your ear. Vibrations start here, and then you have your middle ear bones. And so your stapes strikes the oval window, causes vibrations, and that vibration is passed to the fluid in the cochlea. So you're going further inside your ear. So the vibration passes to the fluid causes hair cells to vibrate. So sound is vibration. Those hair cells are mechanoreceptors, and they release a chemical to the sensory neuron of the auditory nerve. So all of this is based off of vibrations, and the vibrations basically just keep moving through your ear until they hit the hair cells. Once the hair cells are hit, then the hair cells send that message to the auditory nerve, and those impulses are transmitted to the brain for you to understand what it is that you're hearing. So let's talk a little bit more about the hair cells. So the hair cells have stereocilia, which are just tiny hairs that stick out and detect specific wavelengths. As they bend back and forth, you are understanding that your hair is sensing that change, that internal change, and this change is what is producing the impulse that is then carried to the nerve. Now, semicircular canals, these are also located in the inner ear. There are 3 at right angles in different planes. So what that means is they all are at a right angle, but you know how there's the X plane, the Y plane, and the Z plane, in math? It's the same thing in your head. So depending on how your head is moving, they can determine that you are actually moving. So within each canal, you have a cupula, which is just this gelatinous cap, I guess, that covers the hair cells. So you can see the hair cells within it. And then you have this I don't know, dollop of gel that's just over the hair cells. So with movement, the cupula will move, and you can see it okay, I'm moving, whatever, my body is moving, so my cupula is moving in the opposite direction, and it will stimulate the hair cells, and this will cause an impulse to be sent via the vestibular nerve. This is how you know that you are actually moving, and this will get sent to the cerebellum. So this is a slightly different, well, it is not a slightly, it is a different nerve, and it does a different thing. This detects movement, whereas the previous slide was all about detecting noise and sound. But this is what causes motion sickness, if you've ever experienced motion sickness. So the direction of body movement, let's say that you're moving to the left, your cupula will move in the opposite direction because it's being pushed in the other way. But sometimes the reaction of this is a little oversensitive, and so it feels like you're moving significantly more than you actually are. Or on the flip side, if you're in, that would be if you're in a roller coaster or something and you're moving and it's just like freaking out and you're getting like really sick by riding a roller coaster or something, that's because although you're sitting, your ear is portraying that you're moving at a different speed. This also happens when you're sitting in the car. This is where I personally get car sick, I get motion sickness in the car. And the key here is that you are sensing with your eyes that you are moving. But because you're sitting still and it's like a pretty calm road, you're not really moving much, so your body isn't understanding that you are moving, and the disconnect between visually seeing movement but not actually having these semicircular canals show movement is what makes you feel sick. For me, the way to cure that is to just open the window so I can feel the wind, which helps my body sort of realize, okay, maybe there is some movement around here, so I'm not gonna like make you throw up. And then the last thing we're going to talk about today is cochlear implants. If hair cells are defective, but the cochlear nerve is still functioning, you can use an implant. This is for people that are hard of hearing or close to deaf, but the cochlear nerve does still have to be active to have a cochlear implant work. So there are two different parts to a cochlear implant. There are external parts and internal parts. The external parts consist of a microphone, which you can, like, if you've ever seen a cochlear implant, it looks like this on the side of the guy's head. So the microphone detects sounds, and the speech processor selects frequencies used in speech so that you are only getting, the speech sounds versus like ambient noise that's just in the background. And then that transmitter sends whatever the sounds that the processor has processed, it will send those sounds internally. So right around the ear, the piece that like connects behind the ear is the microphone and the speech processor, and then the part that is connected via the wire that goes onto the actual head is the transmitter. So once that transmitter sends those sounds internally, then we go into the internal parts where a receiver is there that picks up the transmitter signals. So it's like right on the other side. You have a receiver picking up the signals. It stimulates the signals and converts it to electrical impulses. And then those electrodes within the cochlea stimulate the auditory nerve. So you're basically like bypassing the ear, like all of the like inner ear bones, the eardrum, all of that stuff, and you're kind of just skipping right over it and doing like a secret shortcut to the cochlea. And then the cochlea will just stimulate the auditory nerve, and then you can hear whatever it is that, or you can like decipher via your brain whatever sound it is that you were processing. Okay, so that's it for this set of notes. We are going to practice a lot more. We're going to do some dissections on this so that you can actually see how all of the parts within like the eye work. We're going to do some practice with actually testing your senses. How well do they actually work? I guess we'll see in class. So good job, and I'll see you later.